Welcome to the deep dive. Our goal, well, is pretty simple. Take complex source material, pull out the core insights, and give you the knowledge you need to be really informed fast. Think of us as your shortcut to those aha moments, often buried in dense research. Today, we're tackling one of the biggest challenges we face globally: the accelerating loss of nature. I mean, the statistics are genuinely alarming. The WWF Living Planet report, it estimates something like a 69% drop in average wildlife populations worldwide since 1970. 69%.
That number is staggering, yeah.
Yeah.
And it creates this huge parallel problem, almost a logistical crisis.
Mm.
We have these international targets, treaties like the Convention on Biological Diversity. They require countries to constantly monitor and report on these changes. But the thing is, the standardized data, the right indicators, and honestly, the sheer infrastructure needed to track biodiversity on land, especially at scale, it just doesn't exist yet.
Right.
Trying to quantify rapid ecological shifts across whole continents, it feels almost impossible, operationally speaking.
It really does feel like we're flying blind when we desperately need, I don't know, pinpoint GPS. But that very idea that we need to build everything from scratch, that's exactly what the paper we're diving into today challenges. We're looking at a really clever study called Air Quality Networks Collect Environmental DNA with the potential to measure biodiversity at continental scales. Lead author is Joanne E. Littlefair, along with her colleagues.
And what's so fascinating, what really gives this study its punch, is the core idea. Maybe we already have the monitoring system running right now, completely by accident, hiding in plain sight. So our mission today is to unpack this concept that the equipment we designed to check for air pollution might actually be the best tool we have for tracking the biodiversity crisis on land. It's a bold claim. Okay, let's get into it.
Let's start with the basic question they were investigating. Can environmental DNA, eDNA, basically the tiny biological traces left by local plants, insects, animals, can that stuff be captured sort of passively as a byproduct on the filters already used by air quality networks?
Exactly. And to test it, they used existing government infrastructure right here in the UK. They looked at samples collected by the National Heavy Metals Ambient Air Quality Network. This network routinely sucks in air, collecting what's called PM10 particulate matter onto these special filters. It's all done for pollutant analysis, managed by the environmental agencies.
Okay, hang on. Let's just clarify those acronyms because particle size seems really important here. For listeners maybe not deep in the environmental science weeds, what are PM10 and the other type they mentioned, TSP? Are we talking like visible dust?
Yeah.
Or something smaller?
That's a great point. So
Yeah.
PM10 means particulate matter that's 10 micrometers or less across. Tiny. About a seventh of the width of a human hair. TSP, or total suspended particulate matter, that's a bit broader, includes some larger stuff too. But the key thing is, these filters are designed to catch microscopic pollutants like lead, nickel, that kind of thing, not specifically designed to collect biological material.
So filters meant for industrial smog and car exhaust. And the researchers are asking, hey, alongside the lead and soot, have we also been accidentally trapping DNA from everything living nearby?
That's the crux of it. And the method they used. Super simple, which is crucial if you're thinking about scaling this up. They didn't need brand new samples or fancy prep. They literally just took quarter portions, small slices of these existing filters that were already collected and just sitting in storage waiting for analysis.
And to make sure it wasn't just a fluke, they compared results from two different setups, didn't they?
That's right. First, they looked at controlled samples. They used a total suspended particulate, the TSP sampler running just outside London. And they varied the collection time. 1 hour, 1 day, and 1 week, just to see how quickly that DNA signal built up. Then they compared that data to historical PM10 samples, remember, the smaller particle size from a site up in Scotland. These were collected over 7 days as part of the network's normal routine.
Okay, so tiny slices of old filters. Filters designed for checking metal pollution, not life. And what they found was, well, pretty mind-blowing. They pulled out eDNA from over 180 different types of organisms taxa. Vertebrates, insects, plants, fungi, the works.
And this is critical. It wasn't just random biological noise.
Right.
The mix of species they found. It matched the known local ecology of those areas. It really suggested the filters were giving a true snapshot of the immediate environment.
Let's make that concrete for everyone. What kind of cool stuff was actually floating around in the air, ending up on these filters? Give us the highlights from this invisible safari.
Yeah, it wasn't just, general animal DNA. They found specific UK wildlife signatures. Things like badgers, dormice, which are pretty elusive little owls, even smooth newts. It's like an invisible audit of who's living nearby.
And it wasn't just the charismatic stuff, right? It also picked up species important for conservation tracking.
Absolutely. They found hedgehogs, which are a species of conservation concern here. And across both sites, they detected something like 34 different bird species, including various songbirds. It shows the method is sensitive enough to potentially track populations we're worried about.
And not just fauna, but flora too, a whole landscape picture.
Exactly. Trees were well represented: ash, linden, pine, willow, oak, and the ground cover stuff too. Yarrows, mallows, daisies, nettles, different grasses. You could even see signs of human activity like crops, wheat, soybean, cabbage, and things we don't want, like certain plant pathogens, fungi like Septoriella. It really gives you a systemic view.
That is an incredible amount of detail from just a bit of filter paper. But okay, does this only work if you're lucky? Or does sampling for longer give you, like, a better picture?
Good question. They looked right at that. Comparing those controlled samples, the 1 hour, 1 day, 1 week ones, they found that longer sampling times definitely increased the number of species detected, especially for the DNA markers used for vertebrates and arthropods. It strongly suggests the eDNA is just accumulating on the filters over time. So the longer you sample, the richer the signal gets.
Okay, so we know what they found a lot, and that longer sampling helps. But now we get to the part that shifts this from just, interesting research to something that could genuinely change global environmental policy. This is the bit that might give us a historical baseline right now.
Exactly. This is about the unexpected finding on stability. Remember those PM10 samples from Scotland collected over 7 days? Sure, but then they were just stored for 8 months at room temperature, no special conditions. And then they analyzed them for eDNA. And they found the DNA was still viable, still detectable, easily.
8 months. Unrefrigerated, just sitting there, and the DNA's still good. Wow. That stability is...
Well, it's kind of astonishing. If DNA fragments can last that long under normal storage, what does that imply for conservation efforts, like immediately?
It potentially rewrites the whole playbook for monitoring biodiversity on land. It means we might already have decades of historical data collected and archived all around the world. Because these air pollution networks, they haven't just been sampling today or last week. Many have been collecting daily or weekly samples pretty densely for decades and unintentionally preserving eDNA right alongside the pollutants.
The potential cost saving, the speed you could implement this, it's staggering.
Yeah.
We might not need governments to find billions to build all new monitoring stations everywhere. We might just need to, analyze what's already sitting on shelves. Precisely. And these aren't just scattered national efforts. We're talking about huge coordinated networks that cross borders. Think about airbase across Europe, Capmon in Canada, the EPA networks in the U.S. They're globally distributed, although, currently more concentrated in North America, Europe, and Asia. But the foundation is there.
So the critical value proposition here is that these existing air monitoring systems could be the best shot we've ever had at measuring land-based biodiversity in a way that's standardized, partly automated, controlled, and importantly, repeatable across sites and over time.
It moves us away from relying solely on, say, occasional field surveys towards something much more like continuous automated monitoring.
And that connects directly to the time series potential you mentioned, which is like the holy grail for conservationists, right?
Yeah.
Understanding change over time.
Exactly. Because the DNA seems surprisingly stable, these stored filters, some maybe going back decades, offer this incredible elusive opportunity to actually track ecological change through time. You could potentially compare a filter from, say, 1995 with one from 2025 from the same location and see the shift, see how the local ecosystem has changed. That's the kind of historical baseline we desperately need to understand that 69% decline we started with.
It's such a powerful idea. But okay, no study's perfect. This research opens up some really important questions about the signal itself, doesn't it? If we're going to lean on this globally, what are the big unknowns we need to sort out first?
Right, there are definitely key research priorities now. First off, quantification. We need to pin down the spatiotemporal nature of the signal. What's the effective range of these samplers? If it detects a badger, was that badger 10 meters away or 10 kilometers away? And we need much more detail on the exact degradation rate of airborne eDNA under different real-world conditions.
And that particle size question comes back too. They showed it works for TSP and PM10, but lots of networks focus on even finer particles, PM 2.5, 2.5 micrometers. Does eDNA stick effectively to those tiny particles too? Because that would massively expand the number of usable filters worldwide.
That's a key technical question, yeah. Then there's the general challenge of detectability. Why are some types of organisms easier to detect than others? Is it about how much DNA they shed, their total biomass in the area, or just how the DNA moves in the air? We don't fully know.
And then there's the foundation of all this DNA work, the reference libraries.
Exactly. Identifying those 180 species in the UK, that relies on having really good comprehensive reference databases like GenBank or Boldy. They're like massive fingerprint libraries for DNA sequences. If you find a DNA fragment, you need that library to tell you which species it came from.
And I'm guessing that's a much bigger hurdle if you try to use this in, say, a biodiversity hotspot in the Amazon compared to somewhere like Scotland, which is relatively well studied.
Oh, absolutely. In places with huge, less documented biodiversity, the reference databases are much sparser. So your ability to identify everything the filter collects would be lower, at least initially. Expanding those libraries for understudied regions is a huge ongoing task needed to make this truly global.
They did mention something positive about the signals range, though, right? That it seems fairly local.
Yes, that's important. While tiny particles like PM10 can travel huge distances, even between continents, the early experiments suggest the actual eBNA signal you pick up is mostly local. And that localization is really useful. It means the filter is likely reflecting the ecology right around the sampler, not just some blurry average from hundreds of miles away. That makes the data potentially actionable for local conservation.
So pulling it all together, what's the big takeaway here? What should happen next?
Well, the conclusion feels pretty urgent. We really need to start investigating the full potential of these existing air quality systems now. That means funding research to analyze more filters, test the methods, answer those questions we just discussed. And perhaps, just as crucially, we need to make sure we start preserving the existing stored filters worldwide. Treat them like the invaluable historical archive they might be before time and ambient temperatures degrade them too much.
It's an incredible thought, isn't it, that an existing infrastructure built purely for pollution control and public health might have accidentally been running the perfect system for monitoring biodiversity across continents for decades. A completely unexpected silver lining just floating in the air.
It really is. Just imagine. All those decades of ecological history potentially sitting right now on dusty filter papers in archives around the globe. What shifts will they reveal when we finally look? How will that long-term record change our understanding of how fast things have really been changing? That's definitely something for you to think about.